Hi everyone, I'm Wan-Chun. I'm a PhD student in Brandon Shad's lab at McGill University. And today I'll be talking about our work on virus targeting and how it acts as a dominant driver of host interfacial evolution in the structurally resolved human virus protein-protein interaction network. That's a long title, sorry. For hundreds of millions of years, viruses and their hosts have been locked in this persistent battle where viruses continuously evolve to bind to host proteins and hosts counter-evolve to disrupt viral binding. So this constant and never-ending tug-of-war between viruses and their hosts is known as an evolutionary arms race. So the focus of our work is to understand the consequences of this evolutionary arms race from the host's perspective. And so we want to know how these virus-targeted host proteins have evolved over time in response to virus targeting. So before we can do this, we first need to distinguish the different types of protein-protein interactions that involve virus-targeted proteins. We have exogenous PPIs and endogenous PPIs. So exogenous PPIs are interactions that occur between a virus and a host protein, and these are often harmful to the host because the virus is using these interactions to disrupt or hijack normal host functions. So I just want to be clear here that since our focus is on the effects of virus targeting on the host, we don't include any interactions where the host is targeting the virus specifically, such as in immune-related responses or antigen recognition. And the second type of interaction is an endogenous PPI. So these are interactions that occur between host proteins themselves, and they're very important for carrying out normal functions within the host. So it's pretty clear here that these two types of interactions serve completely different purposes within the host. So exogenous interactions are very antagonistic towards the host, while endogenous interactions are cooperative. But what's really interesting, though, is that even though exogenous and endogenous PPIs are so different from each other, their binding interfaces actually overlap very frequently. So this interface overlap phenomenon is known as interface mimicry. And the fact that this is such a prevalent phenomenon means that these viruses actually prefer to target the same binding interfaces that the host is already using for its own interactions. So basically, we can effectively split these exogenous and endogenous interfaces into three separate sub-interfaces. The exogenous specific interface that's targeted only by virus proteins. And we also have the mimic targeted interface that's targeted by both virus and within host proteins. So these are the regions where these two exogenous and endogenous interfaces are overlapping. And finally, we have the endogenous specific interface that's targeted only by within host proteins. So from an evolutionary perspective, it's unclear how these three different interfaces evolve relative to one another. But based on our understanding of an evolutionary arms race, we would expect these exogenous specific interfaces to be very fast evolving because the host would want to evolve to disrupt viral binding at these interfaces. Whereas based on evolutionary and functional constraints, we would expect these endogenous interfaces to be much slower evolving because the host would want to maintain its own interaction interfaces. But our main question here is what happens at mimic targeted interfaces? So these are interfaces where both the host and the virus are binding. And in a sense, we technically have these two different types, opposite evolutionary forces at play over here for the mimic targeted interfaces. So in order to answer this question, we first need a framework that can distinguish these three different interfaces, as well as to quantify their evolution. So here we first start out by combining exogenous human virus PPIs with endogenous within human PPIs. So all of this along with 3D structural data to build a human virus structural interaction network where we can zoom in on each of these protein-protein interactions and study their corresponding interfaces at high resolution. So in the next couple of slides, I'll briefly go over our pipeline for building a human virus structural interaction network. But basically, it's broken down into 3 parts. For the first part, we have exogenous interfaces. The second part, we have endogenous interfaces. And then finally, we categorize all these interfaces. So in the first part of our pipeline, we first start out by collecting all the human virus PPIs from INT-ACT. So that's the diagram over here. And we also collected all the protein sequences, so all the human protein sequences and all the virus protein sequences from UniProt. And as well as all the 3D structures and their corresponding chain sequences from the protein data bank. So after that, we mapped these known human virus PPIs to 3D structures based on either existing 3D structures for these interacting proteins, or we use a homology-based structural annotation approach where we assign these interacting proteins to 3D structures based on sequence homology. And also to increase our human virus PPI coverage, we also curated and manually validated PPIs from the protein data bank. So after that, we annotated the interfacial residues on our human target proteins. And these were the residues where the human protein is in physical contact with the virus interaction partner. So these are shown in red boxes over here. And they were defined as residues whose relative solvent accessibility decreased upon binding to the interaction partner. So the first part of our pipeline that I discussed before was for exogenous PPIs. And now in the second part, we do essentially the same thing, but now we take all the human target proteins that we had before, and we structurally resolved and annotated their endogenous PPI interfaces as shown over here. So over here is the resulting human virus structural interaction network. This is a nodes and edges view of the structural interaction network, where each of these exogenous and endogenous PPIs is represented by at least one high-resolution 3D protein complex structure. So in the third and last part of our pipeline, we gathered all of these exogenous interfaces and all of these endogenous interfaces across all of our human target proteins. And then we categorized them into exogenous specific, mimic targeted, or endogenous specific. So the table down here shows the number of residues in each of these interface categories. So now that we've properly categorized each of these interfaces, the next thing we need to do is to calculate their evolutionary rate. So here we use site-specific DNDS as a measure of evolutionary rate. And these rates were based on multiple sequence alignments between reciprocal best hit orthologs from 19 other mammalian species, as shown over here. And specifically for this approach, we have one DNDS value for each residue of our human target proteins. So now to compare the evolutionary rates of different interfaces, for each of these individual interface types, we computed an overall mean site-specific DNDS value across all the residues in that interface type. So the table down here, the plot down here shows these overall evolutionary rates for each interface category. And on the right, we also included these non-interfacial surface residues for a baseline comparison with our interfacial residues. So specifically comparing these interfaces, we see that the exogenous specific interface evolves significantly faster than the endogenous specific interface. And this makes sense because the host would want to evolve to disrupt viral binding at exogenous sites while maintaining its own endogenous sites. But what's really surprising is that these mimic targeted interfaces that are bound by both human and virus proteins actually evolve as fast as their exclusively virus targeted counterparts over here. So it's almost as if there's no endogenous targeting occurring at mimic targeted interfaces. So this clearly shows us that virus targeting is dominating endogenous constraints and driving the evolution of mimic targeted interfaces. So to explain this in a little more detail, if we consider the different interactions that occur at each interface, we can say that the only difference between mimic targeted and exogenous specific and between endogenous specific and non-interfacial residues is this presence or absence of endogenous targeting. So based on this difference in endogenous targeting, we would expect there to be this similar increase in evolutionary rate going for endogenous specific to surface residues, as well as going from mimic targeted to exogenous specific residues. So, but in reality, what we see is that these exogenous specific interfaces aren't as fast evolving as we would expect. So what this tells us is that these mimic targeted interfaces, the ones that are bound by both human and virus proteins, are in fact the key battleground in this host virus evolutionary arms race. So we also wanted to see how far back we could trace these evolutionary signals of an evolutionary arms race between host and virus. So the plot on the left shows our original analysis with mammals. And we conducted additional evolutionary analyses to compare our human target proteins with two more distant lineages. So the non-mammalian vertebrates and the invertebrates. And what we see is that in non-mammalian vertebrates, these mimic targeted interfaces are no longer as fast evolving as they were before. And in fact, it actually looks like they actually start to resemble endogenous specific interfaces. And then if we go further back into invertebrates, we see that even these exogenous specific interfaces start to resemble non-interfacial residues. So this shows us that these virus-targeted interfaces shift very rapidly across different host lineages. And also, in other words, this host virus evolutionary arms race and the precise regions where this arms race is taking place is highly specific to each lineage. So just an important point I wanted to highlight here is the difference between protein level evolution and residue level evolution. So on the whole protein level, viruses actually tend to target more conserved host proteins. So this means that these virus targeted proteins are actually slower evolving than non-virus targeted host proteins. So this was shown in a couple previous studies, and we also confirmed the same results using the proteins in our structural interaction network. But if, as we saw before, if we zoom in on the specific PPI interfaces, we see that these virus-targeted residues are actually faster evolving than non-virus targeted residues. So I just want to clarify here that these two different trends aren't actually conflicting. They're just showing evolution at different molecular scales. So one that's zoomed out on the whole protein, and the other is zoomed in on specific interfaces and residues. So just to summarize everything, we combined PPI networks with 3D structures. So we built this framework that combines these PPI networks with 3D structures to resolve and distinguish PPI interfaces and to overlay residue level evolutionary rates onto these different interfaces. And using this framework, we saw that virus targeting dominates endogenous constraints at mimic targeted interfaces, And we also showed that these mimic targeted interfaces are the key battleground in this lineage-specific evolutionary arms race between hosts and viruses. And also because interface mimicry isn't just limited to viruses, we could apply this whole framework to study other pathogens as well to give us a more complete picture of how pathogen targeting and mimicry together shape host evolution. So with that, I just want to end off by thanking my supervisor, Brandon Shaw, as well as all of my fellow lab members. I think some of them are here. And especially Tingy, Elon, and Leia for lots of helpful discussions, and also funding sources. And I also want to thank the conference organizers for this opportunity to present our work. Thank you so much for listening.
Thank you very much for being such on time. We have time for questions. Any question?
Hello, thank you for the really nice talk. Maybe just a naive question, but because you're evaluating the NDS, so you don't really take into account the physical chemical changing of the amino acid. So can you also consider that in the interface, because maybe it's a positive selection then, but it's not really impacting on the chain.
Right, that's a really good point. So yeah, currently we're using static structures. So you're saying if we look at different conformations, then we can account for different like biophysical, chemical properties. Is that what? The single. Right. Oh, I get what you mean. Yes. Right. So yes, that's one point I didn't touch upon. So right now we're using DNDS as like a general evolutionary rate measure. But as you said, yes, if DNDS is greater than 1, there's positive selection. If it's less than 1, there's negative selection. And then if it's roughly equal to 1, that's neutral selection. I think I had a slide here, but I didn't want to show too much. Let me see. So we did calculate. Oh, whoops. Oh, okay. Let me just do that again. Yeah. Oh, yeah. So this was what we saw before, and we calculated the fractions of residues where DNDS is greater than 1, and the trend holds very consistently across all the interfaces. Yeah. Does that answer your question? Yeah. Okay. Perfect.
We have another question there.
Thank you for a great talk. With your interfaces on the targets, have you looked at population variation from databases like Nomads?
So currently we're not really looking at like the population variance part of this, but that's a future direction that we're hoping to do, where we look at like McDonald-Kreitman tests, where we can, incorporate all that population variance and, population genetics and all that. Yeah.
I have a question.
Oh, yeah?
You said that viruses tend to, at the protein level, tend to target more conserved proteins.
Yes.
This part I don't understand much.
Oh, yeah?
But,
okay.
but the thing is,
yeah.
I don't understand. I mean, I understand what you say, but what's the explanation? Because there's I don't see an evident reason why viruses could not target novel proteins, like for example in higher mammals, for example. I don't know. What's your interpretation of this?
That's a very good question. So this even when I first saw this, I thought it was contradictory, but technically because viruses are very small, right? So they need to find a way to be able to hijack the host. And so the best way to do that would be to target the most conserved proteins. But precisely within these conserved proteins, so specifically within these conserved proteins, you would see these different residue level evolutionary rates. But as a whole, if you look at the entire protein, that protein is very conserved. Does that make sense?
Thank you very much.